Sounds good. All right. Good evening, everyone. So tonight I'm excited to
Beautiful.
introduce a friend and former lab mate of mine and mentor. Got me through my master's. Dr. Alix Matthews, who was originally from North Little Rock, but received her bachelor's in environmental science from Rhodes College over in Memphis before doing her master's and PhD at Arkansas State University, where we met and did her master's work, which led to my master's work. Shout out, Alix. Um, and she's been spending her career studying the relationship between animals and the organisms that live with and or on them. So ectos and endos, um, things from birds and the feather mites that we're going to hear about today, but also worked kind of in a postmasters with ants and the fungi that they farm. She's done a lot of different things. But currently, Alix is a postdoctoral researcher at the University of Buffalo in New York. And I'm sure we're going to get to hear a little bit about what she's been up to up there tonight. So, Alix, take it away. Excited to hear your presentation.
Yeah, thanks. Thanks so much for inviting me too. I'm excited to get to talk about uh some of I mean all of our work on feather mites throughout our time at Arkansas State. So, um without further ado, let's see, maybe. Okay. So just to kind of start off pretty broadly and just to remind or have everyone become aware that birds are, ecosystems of symbionts. There are all sorts of critters living on and on top of and within birds. So, next time you see a bird, you're not just seeing a bird, but you're actually seeing like all of these other organisms that live alongside of it. So, we're maybe familiar with ticks, lice, or sometimes a familiar critter on organisms, viruses, internal parasites. But the star of our show tonight are going to be these feather mites here at the top. Um, so just starting out with what really are mites, because a lot of people, there may be some misconceptions about what mites are, but they are arthropods. So they're within the phylum Arthropoda. They're arachnids as well, so class Arachnida. So they're closely related to spiders, and they're in this subclass Acari. There is this picture here is just a representation of some of the diversity in shapes and sizes of a variety of mites. And this is scaled to the size of a pinhead. So if you can imagine that size. But there's about 50,000 species of mites that are known, which is only about 1 to 5% of the estimated number of species of mites there are globally, which there's the estimates range from like 3 million to 11 million potentially. So there's a really wide range of estimated biodiversity, but only about 50,000 species have been described at this point of mites. And this is all mites. And they are ubiquitous. So they've colonized pretty much every type of habitat, terrestrial, marine, and freshwater habitat. So they're literally everywhere. Um, and one of a quote that I have read before that I thought was kind of puts this into perspective that basically anything that's bigger than a mite has mites. So there are lots and lots and lots of mites out there, all over. So just to give you some another visualization of mite biodiversity and where mites are found and kind of what they look like. So there are soil mites, which I think I think these are pretty cool looking creatures. Pond mites, moss mites. There's also mites really specialized to certain types of habitat. So the one on the bottom left is an ear mite found in rabbits. So there's lots of diversity, lots of habitat specialization, but the presumably the most objectively beautiful mite. I was at a conference on mites in New Zealand a couple of years ago, and when I was talking to people and I was like, oh, I study feather mites, and other people were studying soil mites or, moss mites, they were like, oh, feather mites, these are the most beautiful mites of all the mites. So coming from the mite people, it was pretty exciting to hear that, oh, okay, these guys, I mean, I think they're kind of cute, but hearing that from an objective standpoint was kind of fun. So feather mites, just remember, the most beautiful mites there are. So again, just kind of giving you a broad overview of mites and their evolution. How and when did mites evolve? I found this out pretty recently but I thought it was kind of cool because I'm living in New York right now. So one of the oldest mite, so mites don't really fossilize very well, but there was a mite found in some form of a fossil from the Devonian period found in New York State. So I thought that was pretty cool. This is one of the oldest, probably the oldest mite fossil that we have. But now we're kind of shifting into, I've given you kind of an overview of mites in general, but now we're kind of shifting into, okay, let's talk about feather mites now while we're here. So I just want, if anybody is kind of familiar with the geologic time scale, if you can kind of guess to yourself when feather mites might have appeared. So I just showed you when mites appeared. So, kind of think to yourself, maybe when do you think feather mites appeared? So I'll give you a few seconds to think about it if you have any guesses. But
Jurassic?
yes, well, right in between Jurassic and Cretaceous. Earliest feather mites evolved around 145 million years ago, which is synchronizing with the rise of Archaeopteryx and other flying bird-like dinosaurs. So feather mites appeared about the same time that birds appeared because feathers appeared at about that time. So that gives you sort of a background on the evolution of feather mites. But feather mites, I mentioned there's a lot of organisms that live on birds, but mites specifically, there are lots and lots and lots of different mites that live on birds. So I'll be talking mostly about wing mites today. This is my primary group of studies. So these are mites that live on the wing feathers and tail feathers of birds, the flight feathers of birds. But there's also mites that live inside the quill of feathers. So this is a picture, a microscopic image of a quill that's cut in half. And you can see all these mites shoved up in the middle there. So there's also mites that have inhabited the quill. There's also mites on the top of the skin of birds. These are ones, if you're familiar with what's called scaly leg, these are part of that group. But there's also respiratory mites, subcutaneous mites, downy mites, and others. So birds are just full of mites, and they have these different kind of pockets of environments for different types of mites to live in. But coming back to feather mites, so I have been studying feather mites for over a decade now, and we've been using them as a model system for a number of different questions. So a couple of reasons why we are focusing on feather mites is that they're really common on birds. They're found on almost every order of birds. They're obligate, so they are completely dependent upon their host for survival and reproduction. They're vertically transmitted typically. That's kind of like with an asterisk, because I'll talk about transmission in a little bit as well. But vertical transmission means that they are transmitted from the parents to the offspring at the nest, typically. And they're highly specialized for living and spending their entire lives on feathers. So they have these dorsoventrally flattened bodies that allows them to kind of nestle in between the barbs there. They have these feet pads on the ends of their feet, which allow them to kind of stick to the feathers and prevent being flung off during flight. They're biologically special, so their diet is specialized to what's available on the feathers, and I'll talk about that in a bit too. But besides all of these things, still less than 20% of their biodiversity is known. So they're everywhere, but we know very little about who they are. But given their life history, so their obligate life history, their vertical transmission, their highly specialized lifestyle, there's this presumption that there's this high degree of coevolution between birds and their mites, and this high levels of host specificity. So one species of mite is associated with one species of bird. But after many years of research, and I'll go into this in more detail, but we've seen that there's more host switching and more host generalists than would be expected, given kind of their life history strategies. And their effect on hosts, so their what we call their symbiotic nature, their functional nature, whether they're parasites, mutualists, commensals, somewhere in between is really unclear. So I'll kind of talk about these three main topics: biodiversity, coevolution, and their symbiotic nature, because these are the topics that I've been studying for quite some time. There's other things, if you have other questions that are kind of we've done some side studies on, and we can talk about those later if you have questions about it. But I wanted to kind of give you an overview of how we study feather mite evolution and biodiversity. So this is a poster of sort of just the diversity of, I'm showing lots of pictures of mites, but they really are, they have a lot of morphological diversity. You can see that some of these are really big, some are small, the legs are all in different shapes and sizes. So, you know, there's a lot of diversity within mites, but there's even like an equal amount of diversity within mites that live on bird feathers. So it's actually really amazing. But so here's just a poster of, just to kind of give you a visualization of all this diversity. And in order to, you know, study feather mites, we have to study birds because they're the environment, the ecosystem of upon which feather mites live. So I've been studying mites on warblers. You know, these are a really diverse family of birds in North America. There's about 49 to 50-ish species that breed in North America, pictured here, and they have, they're relatively recently kind of diverged in evolutionary time, but they've had this massive explosion of speciation in terms of their obviously their plumages and morphologies, but also in their ecologies and different life history traits. So they make a really good study system for thinking about biodiversity and evolution and ecology all mixed in, especially in association with symbionts such as feather mites. So, you know, we have to go out and collect feather mites from birds. So we do this primarily using mist nets. So we set up a mist net, which I'm assuming most people in this audience are fairly familiar with, but we typically do targeted netting, where we set up a net, we're targeting a specific species, and in this case, a cerulean warbler. We caught in this net, and then we go out and extract the bird from the net, and then we put a band on it, this silver USGS band, so that we know that we've caught the bird before. In some cases, we also put colored leg bands on the bird so we can identify the individuals later. And then after we take our measurements of the bird, you know, wing chord, mass, age, all that kind of stuff, we then look for these feather mites. And on warblers in particular, a lot of species of warblers, you can see we're just holding these feathers up to the light against ambient light. All of these little dots are feather mites. This is a zoomed in picture of it as well on the bottom right. But so that's what we're looking for. This is the organism. And you know, these birds are really small. You think about the size of a feather. If you've ever picked up a feather on the sidewalk, it's also really small. And then looking more closely at the feather, you see these really tiny little, basically little specks of dust. But these are fully, you know, alive organisms that have their own ecology and their own evolution, and that's what we're interested in. So, typically we either pull a feather to collect these feather mites, or we kind of brush them off using a small brush. So, okay. I think I'm missing. No, not okay. Sorry. So we went out and collected feather mites from. One of our big questions was, do all warblers have mites, and what kind of mites do they have? So we went out over a couple of years and sampled 45 out of the 49 warblers that breed in North America, multiple different species, individuals and locations for each species. And these images are just different pictures from all the different habitats that we were working in. Thinking about working with warblers, we have some species that are bottomland hardwood associated, some that are more kind of piney associated bogs. We have some that live in desert regions, some that live across or along rivers. So just thinking about the habitats and different environments that we're working in, just to give you an idea of that. 38 of the 45 host species had mites, just any mites on their bodies, which equates to about 84% prevalence. And there's lots of variation in mite abundance within and across species. You can ask Kevin about this. This was part of his master's work. There's some individuals have a ton of mites with, some, say cerulean warblers have a ton of mites. Some individuals don't have very many. Cerulean warblers typically have a lot of mites, whereas pine warblers typically don't have very many mites. So there's a lot of variation with abundance across species and within species. But the 7 species that didn't have mites of our 38 of 45, these were all southwestern species. So I have a picture here. We went out and collected mites from southwestern warblers. So this is like the red-faced warbler pictured here, Lucy's warbler, painted redstart, Virginia's warbler, a couple of the southwestern warbler species. None of them had mites. So that was kind of a interesting pattern that we're interested in looking into further in the future, but just to keep that in mind. There's a lot of variation in abundance. So after we collect these mites and kind of get an overview of which host species have mites, how many do they have, our next kind of step in the process is to extract DNA from the mites, as well as hold on to the body of the mite so that we can have a voucher specimen to look at its morphology in more detail. So this is a picture of a mite. You can see it right there next to the pen. That pin right there is about the width of a hair. So these are really, really tiny organisms that we're trying to extract DNA from so that we can understand their evolution and genetics better. So again, just to give you perspective on things, that is how big the mites are. Typically, I will either poke a small hole in it or accidentally poke a large hole in it to try to break open that exoskeleton. They have a hard chitinous exoskeleton that I have to kind of manually break open to help release that DNA. So this is, the body of a mite with a big gouge in it that I've poked with my little pin there. I undergo a couple of different processes in the lab to get what looks just like clear liquid, but there is DNA in that clear liquid. So that's what I'm interested in moving forward in my process. So one of the big questions that I am able to answer with this genetic information is potentially novel biodiversity. So like I mentioned, there's a lot of species of mite, there's a lot of species of feather mite, and most of these are unknown to science. So they are brand new species to science. So what I'm showing here is a phylogenetic tree, or like a family evolutionary tree of different mite species that we have found on warblers. And each of these kind of labels here is some information about the host that it came from and the species of mite. And the names over here are the different groups of mite based on the phylogenetic tree. But so we don't have to go into too much detail about that, but I do want to point out that all of these species that are listed in, or that are highlighted in green, these were all species that were brand new to science, never been discovered before, which was really fun to kind of go through and get to describe a new species, come up with a new name. And if you're really reading these Latin and Greek names very well, you might see one that looks relatively familiar to you from Arkansas. We named one of our new species of feather mite after Jonesboro, where we collected the specimen. So it's probably as far as I know, it's the only species on Earth that's named after Jonesboro, which is pretty cool. But so that's kind of fun. But so these are new species. This one here in the pink is a species that we're still working on describing, but it's from a black and white warbler. Again, we compare these to a database that exists and there's no other species like it. We look at the morphological keys, kind of like what we do in field guides, and there's no other species like this. So this is a new species to science based on its genetic data as well as its morphological data. So what we'll do next is kind of take these high resolution images from those specimens that we saved from our DNA extractions and go through and start measuring these different morphological traits, like these little adenal discs or these little hairs, and take all these really fine scale measurements to say, okay, this one is significantly different from its next closest relative. So that's the one way that we get to go through to describe new species using both genetics and morphological evidence. And one thing that I like to show with this phylogenetic data is that, especially with people that are familiar with birds, is that we had a really cool or unique case study in which the feather mites were providing evidence of the taxonomic placement of hosts. So yellow-breasted chats used to be in the family Parulidae along with the warblers, so they used to be grouped with the warblers. Now they're in their own family called Icteridae, which is right in between Parulidae and Icteridae. So right in between warblers and blackbirds lies yellow-breasted chats. And we happen to sequence some mites from yellow-breasted chats. This is kind of a larger phylogeny over here on the left that I have made for another purpose, but we got interested in these yellow-breasted chats for a number of reasons. But I'll point out that in the purple box here is all of the warbler mites from this specific genus of mites. So all the warbler mites are in the purple box. We had a blackbird mite in the orange box. And if you can guess where the yellow-breasted chat mites landed was right in between the two. So the mites were supporting that change or that movement of yellow-breasted chats out of Parulidae as well. So it was kind of cool to see that mirrored image of the mites supporting that movement out of that family. Okay, so getting back to our warbler mites here, again, this that same phylogeny that I was just showing you, but I'm going to point out some other interesting things that we started to see with just based on our examination of the patterns of the phylogeny. So the hosts that all shared this Amarocdectes ischios, we started to notice, okay, a lot of these species are all canopy nesting or put their nest above ground, typically in a taller tree or shrub. Whereas all of the hosts that shared this Amarocdectes seiurus, ovenbirds, Kentucky warblers, Nashville warblers, we thought, oh, these are interestingly, this is kind of just a weird little pattern that we were noticing. Okay, all of these guys place their nest on the ground or low to the ground. Whereas Amarocdectes protnotaria, this was a unique species found on prothonotary warblers, which also have a unique nesting ecology in cavities. So there's a lot more to do with this, but we were kind of interested that mites are transmitted typically vertically from parent to offspring at the nest, but we see that some of these host species, some of these birds are sharing a mite, but they're also sharing the same type of nesting strata or nesting habitat. So maybe there's some horizontal transmission happening across species based on where they place their nests. I'll get back to more on mite transmission in just a second, but basically what the point is that we're finding evidence about their transmission from their genetic data and taking a comprehensive look at the family. So we also, this is just another way to look at this phylogeny is based on their hosts, the mite's host specificity. So whether they are specialized to a single host, like the prothonotary warbler is the only one that this mite is found only on prothonotary warblers, whereas this one Amerdictyceras is found on 3 species of bird. So we're starting to see some variation in that specificity versus this Amerdictys ischios, which is found on a ton of different species, that's 17. So just taking that genetic data, we're getting a lot of information about the ecology of the mites themselves, that there's variation in probably their transmission as well as their specificity, and starting to, be able to ask more sophisticated questions about their ecology just based on what we see here in this single phylogeny. So it's been really fun kind of getting to ask all these different questions just based on this information. But thinking about their specificity, so specialists that are specialized to a single host versus generalists that are generalized on many different host species, we were curious about if there's any differentiation in the kind of population genetics of those mites dependent upon their specificity. So if you've done 23andMe, it's kind of a similar, what we're asking is kind of a similar question where, okay, what's the demographic history of these mites based on their specificity. So we expected these specialist mites that are only found on prothonotary warblers to have, the populations from different populations. So we studied populations of mites from prothonotaries in Arkansas, Ohio, Wisconsin, Virginia. So we were looking at all these different places, and we thought that each different population would have its own kind of genetic signature, just like you would see in a kind of 23andMe result. Whereas a generalist mites, because they're generalized on many different host species, we expected all those populations to kind of have an overlap of genetic structure, or all the populations to be genetically equivalent to one another, no matter where they came from. And I'm going to explain these graphs, but what we saw was completely opposite of what we expected. So these graphs are kind of 2-dimensional plots of a bunch of different genetic data. But the main thing is to look at the colors. So each of the colors represent different locations, different populations of mites and their associated hosts. And then the points are representing individual samples, and whether or not the points overlap with one another is telling us how much genetic overlap there is. So for instance, all of these 3 different populations, Arkansas, South Carolina, and Wisconsin, they have a ton of overlap, and we can't tell any difference between their genetics. Whereas these over here on the right side, Wisconsin, the Ozarks, Pennsylvania, they're all structured. There's no overlap between these circles. So what this is saying is that the specialists have a lot more overlap in their genetic makeup, and the generalists have more structured populations, which is completely opposite of what we expected, given that the generalists are, moving around from host to host. We kind of expected these graphs to be switched. But getting back to the transmission question, using our genetic data, now I'm showing these maps of prothonotaries and ceruleans where the points are showing where we caught the birds and collected these mites. And again, the prothonotary warbler has the specialist mite, and the cerulean warbler has this generalist mite. But what we saw these weird results. We were like, is that, these sort of unexpected results where the generalists had these more structured populations, or the specialists did not. But when we started thinking about it, vertical transmission is happening up here in the pink, in the breeding range of the birds. Transmission of the mites is happening from parent to offspring at the nest on the breeding grounds. But birds are really only spending a small proportion, if you think of this as a calendar, a small proportion of the year on the breeding grounds. The rest of the time, they're either migrating on the non-breeding grounds or migrating again back to the breeding grounds. So there's a lot of time where transmission could be happening outside of those breeding grounds. And what we found based on our mite population genetic structure graphs that I just showed you, as well as some host migration data. So we put geolocators on these birds as well to try to figure out where they were going during the non-breeding season. What we found is that prothonotary warblers typically all go to the same kind of relatively small region in northern Colombia. No matter where they breed in North America, they're all going primarily to this small area in South America. Whereas the cerulean warblers, they kind of have this parallel migration pattern where they, on the eastern side of their population, they're going to this more northern area in South America versus the western side, they're kind of going into the southwest area along the Andes Mountains. So we're seeing sort of differences in their migration, differences in the genetics of their mites. And this is just, I'm just showing you these graphs again, but what we're thinking is happening is that transmission may also be happening on these non-breeding grounds as well, because we found so much overlap with the specialist mites, and the prothonotaries are all going to this one area, which could provide an opportunity for mites to switch, as opposed to switch across individuals from different breeding populations, as opposed to the generalist mites found on cerulean warblers. They never come in contact with one another on the breeding grounds nor on the non-breeding grounds. So that could explain why we have this like variation in population structure. So moving on beyond just sort of the genetic side of things, I mentioned we're interested in mite biodiversity as well as mite coevolution and host specificity, which I've kind of given you a taste of. But that third question that I'm asking is, are feather mites good or bad for birds? What is their impact on hosts? And we don't really have a good grasp on this question, the answers to this question. Most studies, including some of my own, have looked at mite abundance as sort of a proxy to the degree of impact that they have on a host. So if there's more mites, they're having more of an impact. If there's less mites, they're having less of an impact, versus various host condition or fitness proxies. So host mass, host reproductive output, host survival, that kind of stuff. So what the majority of research has looked at, kind of the correlation between those. So if you have evidence of this negative correlation between host fitness and mite abundance, so more mites is associated with a lower level of fitness, that's suggestive or has been suggestive of a parasitic relationship. Whereas if you've got a positive relationship, more mites is kind of correlated with a higher degree of fitness for whatever proxy, that's suggestive of a mutualistic relationship. And if there's no relationship between mite abundance and host fitness, then that's suggestive of, oh, maybe they're not really doing anything, they're commensal. But this is just correlative evidence. There's studies from, there's many studies that have found all, different correlations and looking at different things, different fitness proxies and finding all sorts of different things. So there's not a lot of consensus on the effect of mites based on this type of study. So we decided to do an experiment where we could control the presence or absence of mites, to try to better understand what their impact is on hosts. So we were also interested in doing this in both the specialist and generalist mite as well, because it's an interesting kind of life history trait with especially with these mites on warblers, that we have such this high degree of variability in their specificity, that we were curious if maybe specialists and generalists had different impacts on these hosts. So we had two, or kind of a 2x2 experimental setup. Again, we focused on cerulean warblers and prothonotary warblers. These are species that we were kind of doing a lot of work on anyway. And we had field sites already set up for these species, and it just so happens that they had sort of this variation in host specificity that we were really interested in. So we had a control group of both the ceruleans with mites, the generalist mite, and prothonotaries with the specialist mite, where we didn't modify or manipulate the abundance of mites. We just left them as they were. Then we had a treatment group where we removed mites from the host. All the mites on the wings, wing feathers and tail feathers. And we caught these birds, we banded them, we color banded them, we measured their mass, their age. We then went back the following year to estimate their survival. So whether or not they came back to the breeding grounds the following year. And we did this for 2 years for both of these groups of birds. And then we ran a lot of statistics to say, okay, did our treatment impact their survival of host or their change in their body condition? And what we found was that treatment had really no effect on host survival or the change in their body condition, regardless of specificity. So both mites showed no pattern of either in either direction. So this is evidence that, experimental evidence that mites are commensals in the context of host survival or body condition. But, we can look at this, we can think about the impact of mites from another perspective as well. So another study that we did was looking at how mites interact with the feather itself, and if mites are actually doing something specific to the feather that could be impacting the birds as well. So, mites live and eat on feathers. And so one of our questions was, are they providing a cleaning service for birds just based on what they are consuming off of feathers? So this is a microscope image of a feather mite, and then I've outlined this clump of basically its lunch. So whatever it ate right before we put it on this slide is kind of what we're interested in. What are mites eating and from the feathers? But not only that, so we're interested in what they're eating, but we're more interested in what they're actually selecting to eat. So if you go to a restaurant or a buffet, and there's all of these options for you laid out on the table to eat, what are you actually consuming? So that's kind of the same question that we were asking of the mites. Mites have this whole feather of microbes and fungi and bacteria and all sorts of things that are available to it to eat on the feather, but what are they actually picking and choosing to eat? So because this might have an impact on their function on the host. So if they, for example, prefer to eat, if they choose out of everything that's available to them, if they are preferring to eat these feather degrading microbes, or sort of microbes that are bad for birds, while they're avoiding antibiotic microbes or microbes that, if left on the feather, are good for birds, then that could suggest that they're mutualists or actually helping birds. But if this is flip-flopped and they are avoiding those feather degrading microbes, if mites are avoiding the feather degrading microbes and leaving those on the feather and preferring to consume the antibiotic microbes, the good microbes that are good for birds, that could suggest that they're parasitic. So we're asking a couple of different questions here. You know, what is their dietary selection? So what are they consuming versus what's available to them? Are they eating whatever's available to them? Or are they picking certain things? And then secondarily, does that impact their hosts? So this is a little figure. We studied this in in prothonotary warblers. And this is just sort of an outline of our study where we were looking at both bacteria and fungi that was consumed, found inside the bodies of mites. What did we find? You know, like that picture I just showed you, what are they actually consuming versus what bacteria and fungi are available to them on the feather. So we're kind of comparing these two environments. And what we found is that mites are really picky eaters, based on a couple of different metrics, but this was kind of our takeaway here. So this is showing a measure of diversity, of bacterial diversity. So we found higher levels of bacterial diversity on the feathers versus what was inside of the body of the mites. And each point is representative of an individual sample. And the lines connecting the points are showing samples that were, you know, mites from that individual feather, so connected by the same bird. But what this is showing is that mites had much lower diversity of bacteria inside their bodies than what was available to them on the feather, suggesting that they're really picking certain things. And this is another kind of way to view that same information, basically showing that feathers, each of these dots is a sample, and basically the spread of dots is showing how kind of the composition of bacteria that was found on the feathers. So a wider spread is equivalent to a wider diversity of bacteria. So we found a wider diversity of bacteria on the feathers than we did on the mites, which have a much more restricted area in this kind of two-dimensional space. And we looked at the specific taxa or species of bacteria that mites were consuming, and we found that several of the ones that they were actually picking to consume are feather degrading, which is suggestive that they are actually cleaning feathers and providing sort of this mutualistic service to birds. So they're both picky and mutualistic in this context. So, you know, just thinking about their effect, it's probably somewhere between, if I were to kind of summarize it, it's probably somewhere between commensalistic and mutualistic. And some of my future directions, or the stuff that I'm working on now at the University of Buffalo, is kind of expanding this microbiome study. So I mentioned that birds have lots of different microbes on them, but they also have different compartments of microbes. So we were mostly focusing on feather microbes and feather mite microbes, but birds also have gut microbiomes and uropygial glands that have their own microbiome that are probably very important for related to feathers, because birds squeeze the uropygial oil out of their preen gland and lay that on their feathers. So we're kind of interested in what's the connection between those two microbial communities. And this is kind of a busy figure, but these are kind of our main objectives, is to look at, does the presence of mites impact the sort of stability of their microbiome? You know, is having mites kind of stabilizing your microbiome or not? We're also interested in the transmission of microbes between all of these different compartments to try to figure out what role mites could be playing in it. And we're also interested in that question of host specificity again. So, the study that I just showed you about mites being really picky, this was based on prothonotary warblers, which have that specialist mite. So just by its specialist nature, it may be more specialized to certain microbes than maybe a generalist would. So we're kind of interested in that dietary specialization related to their host specialization. And some other things that I'm also working on in this lab now that I don't have a specific slide for, but that I can talk about because it's related to this microbiome. We're also interested in how the microbiome could be playing a role in feather coloration. So there's carotenoid producing bacteria. So we are interested in to see if birds can kind of somehow use that bacteria that is producing carotenoids inside of their gut, and if they can kind of sequester that or hold on to it or use it for the carotenoids that they need to color their feathers. So I can talk more about that if you're interested as well. But just, circling back to this is that birds are an ecosystem of symbionts. So, I've mostly been talking about mites and a little bit about microbes as well, but there's lots of organisms that live on birds, and they really are an ecosystem in and of themselves. So when you're looking at a bird, you may look at it a little bit differently now after hearing about all this. So thank you for inviting me, and I'm happy to take questions, and I just want you guys to remember to stay curious and ask questions, because this picture, I like this picture. This was the first picture I took of feather mites. This is just in the lab. We had no idea what they were doing on these birds, and just by asking, like, what is this, we've gotten to answer all of these really cool questions, and so much more to come. So that's all I have, but thank you guys so much.
It's so good to see the Audubon Society's logo there, Alex. We are proud of you.
Yes, definitely. That's been a huge help.
I proudly uploaded all your papers on your website. So for those of you watching, if you want to see any of those, I emailed my students some of your papers, but
Okay,
all your
great.
papers are in the Audubon website.
Oh yeah, great. Yes.
So I have one question. It's
Yes.
amazing that you were able to recapture birds the subsequent summer after they wintered in South America to assess how your experimental treatment affected them. That must have taken a lot of work. How many birds did you recapture?
Yeah, we caught each season, we caught 20 birds, 20 prothonotaries and 20 ceruleans. And then the next year, same thing, 20 and 20. So 40 birds total of each species. And then recaptures, it was just who came back. So we spent a lot of time searching the area for returned birds. And
based on color bands.
Based on color bands, yeah.
Oh, and then you put nets to go after them.
Yes, yeah. So then we targeted those specific. We've kind of narrowed down its specific territory, and then put a mist net in the middle of the territory and targeted that individual after that.
Oh wow! I thought they were random recaptures. No,
No,
I get
yeah,
it.
these were the exact same individuals from one year to the next. So we were able to assess survival of that individual, and then the change. Our metric was body mass. So the change in body mass from year 1 to year 2 at an individual level.
Wow.
Yeah,
amazing.
it was recaptures are really tough.
Yeah, that's what blew me away.
Yeah,
that's great.
warblers are a little bit easier than some species, but they're still very tough to recapture.
Yeah, Cannon, I think from my study with warblers and mites as part of my thesis, we had about 40% of our warblers return out of 120 something. And most of them returned within 75 meters of their breeding season the last year. So they have pretty high pretty high sight fidelity if they are able to survive that full year. So
after spending the whole winter 1,000 miles away.
Yeah, it's pretty amazing.
And it's just like pre-programmed in their brain.
Yeah. And I mean, in some cases they come back to the exact, like with Prothonotarius, we had nest boxes out, and so some of them come back to the exact same nest box from the year prior. So it is really amazing that they
stunning. That's absolutely stunning.
They did not bring the same mites back with them, though, probably. Mites don't live that long. But
yeah, probably not.
They're kids, they're grandkids or something like that.
Yeah.
Alex, Barry put a question in the chat that I'll read.
Okay.
Is there any evidence that things like bird feeders, bird baths, and other points present feather mites with an opportunity to switch their hosts?
That's a great question, and one that, I think mites can move a lot quicker than it seems. So I think any skin to skin contact is an opportunity for mites to jump from one individual to another, and that could include like within hosts or within a species, but also across species. So I don't think there's been any studies on that specific question, but that would certainly be a point of transmit, like a moment of transmission that we could pinpoint down. And there's a lot of questions like kind of related to that, or like roosting sites along during migration. If different species, you've seen pictures of fallouts, and there's tons of birds just like of all different species all piled basically on top of each other. I think that's another opportunity for transmission.
Or bird
Alex,
banders.
I'm looking at
Sorry,
Kevin.
Oh, I was just gonna say, or bird banders or researchers catching multiple birds in a net at once.
Yeah. Yeah, that I think that's probably a lower risk, but definitely a risk.
I'm looking at the list of my students who are attending right now, and I see most of them are interested in grad school. And in fact, at least a couple of them are interested in your own former lab in San Boises and Jonesboro. So what advice would you give them, Alex?
Just in terms of finding or thinking about grad school?
That's the number one concern, finding a good grad program.
Yeah, I think with grad programs, it's really important to have a good relationship with your advisor, or at least a good working relationship, because this is somebody that you're going to be working with very closely especially for a master's program, 2 to 3 years. If you do a PhD, it could be 5 to 8 years of your life is being spent working closely with someone. So I think reaching out to them beforehand is definitely what you should do, and try to contact them early, relatively early for the next year. So like if you're interested in going to grad school next year, right now is a good time to talk to potential grad advisors, because they're thinking about, what funding they may have, or what, somebody that might be graduating from their lab, and so spots opening up and that kind of stuff. So definitely reaching out early and coming with your own ideas is also a good thing. People are interested in having people in their labs that have, their own ideas and are interested in taking a question and making it their own. So definitely doing your reading on the labs that you're interested in, and just trying to strike up a conversation and make sure that you are kind of have a good connection with the potential grad advisor. And also talking to the current and past graduate students is also a really good way to gauge whether who you're interacting with is who you think you're interacting with.
Good advice.
Yeah.
I also noticed NSF on your acknowledgement. Now, of course, all that is being decimated Government funding is almost completely dried out for wildlife studies.
Yeah.
So your advice is even more important to bear in mind.
Yeah, definitely. And the NSF on what I'm referring to here are scholarship programs that have been funded through NSF, not necessarily like my own NSF funding. So grad school, if you're able to find a scholarship, there's within Arkansas, there's a couple different scholarships that you should always apply to. Arkansas Game and Fish is one of them. I'm assuming they still have that conservation scholarship program. But some, master's programs have in Kevin's case as well. We were on a scholarship funded through NSF. So those are another good thing to ask when you're thinking about grad programs as well. But funding for these projects can be variable. So it's always a good idea. You're never going to get any funding if you don't apply. So it's always a good idea just to apply for stuff, and don't be afraid of rejection and not getting it. And you can always learn something by writing about your ideas and writing about your project and trying to pitch it to someone, no matter what. So even if you don't get the funding, you're still getting something intrinsically out of it. And you've got a draft for your next proposal already.
Yeah, absolutely.
Alex, did you find out anything about Lucy's warbler mites? I think they're the other cavity nester, right?
Yeah, good to see you, Jacob. No, Lucy's warblers did not have mites.
Oh, that's right.
So they're one of our 7 southwestern species that did not have mites, which I was really surprised about that whole thing in general, that there was 0 mites on any. We caught a few non-warblers too, and none of them had mites either. So like 0 birds in general out there.
Something to do with desert, something to do with humidity.
It's gotta have something to do with that. We went during a like extremely dry year as well. So I'd always like to go back and ensure that we can, we found what the true pattern there. But yeah, but if anything, if we were to find mites, I was really curious if Lucy's warbler and Pythonotary warbler had the same mite. But
Yeah.
no mites to compare.
Thanks.
Yeah.
So I have a question. What made you interested in studying mites on feathers?
Yeah, that's a very this picture that I'm showing, basically, well, more specifically, I was studying in undergrad. I did undergrad. I was in a research lab as an undergrad between my junior year and senior year, and into my senior year. So I did a little bit of undergraduate research on avian malaria parasites, so blood parasites, on songbirds, just lots and lots of different songbirds. Kind of a lot of the same sort of type questions that I've been asking with these mites. But I had sort of a background on host parasite relationships that I was really interested in still in grad school. And I thought I was coming into grad school for something completely different, and the funding fell through. So we had to come up with a new idea. And then basically we pulled this feather out of a drawer, more or less. And we were like, well, do you want to study this stuff? Whatever this is. And I was like, yeah, I think we can come up with something cool about, this based on my past research with avian malaria parasites. So
was that also in Jonesboro?
That was at Rhodes College in Memphis.
Oh,
yeah. But that kind of sparked my interest in host parasite relationships. And then when this mite opportunity came up, I was like, actually, that's really cool. And basically haven't let that go since then. Still studying mites, and still want to keep studying mites. There's so much to learn about them.
Well, you're becoming a mighty successful scientist.
Thanks. They are mighty cool.
All right. Any other questions for Alix? Either feel free to unmute or put it in the chat.
Could my students contact you if they are interested to follow up or?
Yeah, definitely. I'm sorry I didn't put I usually put my email on here, but absolutely I'm easy to find online.
Right.
So yeah, absolutely. If you guys have more questions about mites or birds or graduate school or Arkansas State or anything, feel free to contact me. I'd be more than happy to chat.
Thank you. That's
Yeah.
great.
I'm glad you all came. I hope it was interesting enough for it. Well, it's 9 o'clock here. It's only 8 o'clock there, so not as late.
Yeah. Thanks, Alix. I hope everyone enjoyed it. And Alix is a great example of what a little bit of Arkansas Audubon Society Trust money can do.
Yes,
Go and
definitely.
Absolutely.
do great research and get even more grants. And yeah, Alix is a wonderful human too. So that's also great. So
As is Kevin. This is a great group here. I'm really glad to have been a part of it tonight.
Yeah, we appreciate
Thank you,
it.
Alix.
Yeah, thank you guys. And all the emojis.